In 1961, scientists Nuremberg and Mattei began working on determining the mostly universal genetic code. From previous research, they knew that mRNA mixed with ribosomes and tRNA would create proteins, but they did not know how these proteins were coded for. They began an experiment with a mixture called a cell-free extract from lysine and E. coli cell. This contained the ribosomes and molecules needed for translation. The mRNA that they also added to the test tube was a special homopolymer known as a poly-U chain, as it was made up entirely of U's. To figure out what amino acid would bind to what codon, they added 19 normal amino acids and a tRNA molecule with a radioactive version of an amino acid attached to it into the test tube. They let the mixture incubate at 37 degrees Celsius to allow translation to occur and to produce the protein chains. With this process, if the radioactive tRNA matched the codon on the poly-U, the resulting precipitating protein would be radioactive, and if it didn't match, there would be no protein product. Nuremberg and Mattei had to repeat this experiment 20 times, changing the radioactive amino acid each time until all were tested. They found that the codon UUU only ever produced a radioactive protein chain with the amino acid phenylalanine. They repeated this experiment with the homopolymers poly-A, poly-C, and poly-G, finding that AAA coded for lysine, CCC for proline, and their experiments with poly-G never worked. The next advancement in genetic code came a few years later in 1964 with Nuremberg and Lieder. They were able to synthesize short trinucleotide mRNAs with known sequences to get all of the combinations of codons possible. With this technique, they were able to perform their experiment, the triplet assay. This experiment worked much like the first and involved filter paper made of nitrocellulose. The paper would allow unbound tRNAs to pass through but would catch ribosomes. Also like the first experiment, they used a singular radioactive amino acid each time, having to go through 20 experiments for each mRNA sequence. If the radioactive tRNA bound to the mRNA in the ribosome, it would be stuck to the filter paper and would be able to be detected. With this technique, Nuremberg and Lieder were able to decode the rest of the 64 codons and their amino acids, giving us the universal genetic code we use today.